This is Greg Stalker presenting information about the aerobic production of ATP. The first breakthroughs in this area of research occurred over 70 years ago, so let's go back in time and start from the beginning. This is German scientist Hans Krebs. Born in 1900, Krebs was only 37 when he discovered the cycle of metabolic reactions that were responsible for the aerobic oxidation of energy substrates. He labeled it the tricarboxylic acid cycle, or TCA. It is also known as the citric acid cycle. It was formally named after him as the Krebs cycle upon receiving the 1953 Nobel Prize in Physiology or Medicine. Krebs shared the honor of the Nobel Prize with this man and fellow countryman Fritz Lippmann for his discovery of coenzyme A. A derivative of the B vitamin pantothenic acid, coenzyme A forms with the acetate ion after macronutrient catabolism, making acetyl-CoA, which is the fuel for Krebs' cycle. Together, these two men opened the door to decades of research, which continues today. This illustration maps protein's contribution to the aerobic production of ATP. Once hydrolysis breaks the peptide bonds, single amino acids are revealed and fill their roles as needed. While branch-chain amino acids can be directly oxidized in skeletal muscle by transamination, other amino acids have to be transported to the liver to be deaminated before undergoing gluconeogenesis. Some aminos can directly enter and serve as Krebs cycle intermediates. Depending on the intensity and duration of exercise, along with the state of nutrition, protein serves as a small contributor, approximately 3 to 18% of the energy substrate used in the aerobic production of ATP. The major contributors are glucose, glycogen, and triglycerides. Glucose and glycogen enter glycolysis, ending in the formation of pyruvate. Pyruvate is then converted to acetyl-CoA by an 8-enzyme pyruvate dehydrogenase complex. Lipolysis breaks triglycerides into glycerol and free fatty acids. Beta oxidation then takes those fatty acids and splits them into two carbon sections to form acetyl-CoA. Taking place in the mitochondria with acetyl-CoA as its fuel, Krebs cycle contains 11 reactions but only produces one ATP by substrate phosphorylation of GTP in the 8th reaction. GTP then donates a phosphate to ADP to form ATP. The primary role of Krebs cycle is to finish the oxidation of energy substrates, produce carbon dioxide, and produce the hydrogens that will be used in the electron transport chain. Embedded in the inner mitochondrial membrane is a series of proton pumps, cytochromes, and respiratory assemblies. Reduction-oxidation coupling moves the electrons across the cytochromes, activating the pumps that transport the protons from the mitochondrial matrix to the intermembrane space. Once a concentration gradient is established, the protons diffuse back to the matrix through respiratory assemblies, activating ATP synthase and the reformation of ATP and water. This process is known as oxidative phosphorylation and is the primary source of ATP production during rest and submaximal exercise. This illustration displays the total ATP production from one molecule of glucose. Due to the consumption of 2 ATP during glycolysis, the net production is reduced to 32 ATP. Unseen is the enormous capacity of triglycerides as an energy reservoir that yields a total of 463 ATP from one 18-carbon molecule. Oxidative phosphorylation is an ancient form of energy production that covers the entire biological spectrum, from single-cell organisms to the largest beings on the planet. Without it, life would be unable to exist as we know it. That's why the science and the process behind the aerobic production of ATP is truly a marvel.